Yeah, sure. All right, so let me get this up here. And here. Okay, so that I don't have anybody to interact with in the room. So anybody who's called in or online, please feel free to interrupt me because I don't have any feedback about how this whole thing is going at this point. I'm just staring at a screen in a room. So welcome. The reason that we put this presentation together was actually as a specific request from some people who were seeing all of the new things coming out of NIH about new initiatives and new changes to the grant process. So what I want to do is give you some background about why these changes are being made, and then what that really means to you when you're putting together your application for support. So I just want to start saying that if anybody who's on the call, if there's any other particular topics that you haven't seen covered in any of these grantsmanship courses should happen to be coming up, please feel free to email Erin and we can put them on the agenda for future workshops. So this presentation's about the two new criteria that were added to the NIH grant mechanism in 2016, and that is the whole concept of evaluating scientific premise and these new initiatives related to rigor and reproducibility. So what I want to go through is why NIH is making these changes. And what this really comes down to is something that some people have termed the reproducibility crisis, has met a concept that anybody who's ever worked in the federal government in Washington would call the Washington Post test. So the reproducibility crisis was really in this flurry starting about 5 years ago. There were multiple high-profile reports of published scientific studies that were shown to be irreproducible by others or of questionable value due to misidentification of the biological material. And the earliest of these comes out about 10 years ago, but really in the past 5 years, this has become extremely high profile. Now, from the standpoint of the Washington Post test, if you work within the Beltway, that very often there has to be the perception that you're doing something about a problem. And that if you're doing something in the government, could it survive being on the front page of the Washington Post? And there are decisions that are absolutely made by how does the appearance of what's going on really matter. And we'll get into how all this is playing out in a minute. Now, one of the very first high-profile irreproducibility problems came from the misidentification of cell lines. So for those of you who are on the call who aren't benchtop biomedical researchers, cell lines are basically cultured cells that are coming from a whole host of diseases or biological materials that are maintained in culture for days, weeks, and actually at this point, decades. The HeLa cell was the very first of these cell lines, and it is extremely highly aggressive cervical cancer cells. It is so highly aggressive that a single cell contaminating a culture can take over from a normal cell line. So, and that it seems that even a very small amount of culture volatilization can allow for a single cell to seed a new culture, well, it will take it over. And there are a number of published reports where this has actually happened, including a case of a study that was supposed to be normal liver cells, which turned out to be done on HeLa cells. And liver cells actually go very slowly in culture. The lab was culturing HeLa cells at the same time. The culture got contaminated and nobody realized it. Another case was HeLa cells being non-transformed amniotic epithelial cells. non-transformed intestinal epithelial cells, etc. And particularly a lot of work on normal cell lines has been contaminated by these cell culture problems because normal cells grow very, very poorly in culture, while these aggressive cancer lines can easily cross-contaminate. In my field of vision research, there was a retinal cell line that was used in hundreds and hundreds of papers. And when they went back and actually analyzed it, they realized that the cell line that everybody was studying was not, in fact, retinally derived. And they were able to go back to some original cultures. And it turned out the original retinal cell line never even left the lab that developed it. It was contaminated extremely early in the development of the cell line. And there are numerous examples of this. So that's why there is a really big push now about validating biological material, because there's so many high-profile publications where it turned out that the biological material was in fact contaminated very early on. And there was also some very high-profile studies coming out of drug companies, and these are two. Drug companies really mine the basic preclinical biological and biomedical literature. What they're looking for is potential leads that they can develop into drugs and bring into the clinic. But these drug companies have learned that they have to, in fact, replicate the main findings in their own lab. And Amgen reported that they could only replicate 11% of 53 preclinical cancer studies that they were interested in. Bayer Pharmaceuticals could only replicate 20 to 25% of the preclinical studies they tried to replicate in-house. Now, and there have, in fact, and this is really a bottom line issue for these drug companies, because there have been many cases of failed clinical trials and other research studies that failed because of an assumption the published data is fact. And to even give you an example that maybe might come a little closer to home, even at the standpoint of individual labs. When I was in graduate school, one of my colleagues took over a published project and was working on the next steps in the finding. When she was a fourth-year graduate student, her committee said that she really should validate and replicate the original finding, and she never could. And this meant that she no longer could get a degree. And actually, in my own laboratory, I make sure that if I have a new graduate student joining my lab, I always make sure that they primarily replicate the finding that their project is based on. And these are even studies that are coming out of my own laboratory, because I don't want a student to have many, many years of work in when what they're studying, they couldn't even basically replicate. And I know there's a lot of people who are involved in this network that are really doing psychology, social science, community engagement research. And that field has also its own problems. That there is a study that says that people tried to replicate 98 original high-profile psychology studies, and they couldn't replicate over 50% of them. But to show you how tricky this whole problem was, is that there was another study that suggested that the replication attempt was in fact fundamentally flawed, because they had methodological errors on how they chose to replicate the data. And it also is, replication is often not an all or nothing thing. Is it you completely replicate the data? Do you get the basic gist back? 10% of the data is replicated, and so forth. So it basically, all of these studies have been coming out and have really suggested that there needs to be some really hard looks at this idea of whether data collected is reproducible, and whether the study design and the biological resources coming into the study are in fact valid. And what all of this comes down to is that it is extremely easy for investigators to fool themselves. While, of course, there have been very high-profile cases of scientific misconduct, in that it's not believed that most of these replication problems are in fact due to direct scientific misconduct. It has to really a lot to do with intrinsic biases. Some of it is that that we really have some big conflicts of interest. Those of you who do clinical research, if you go to a clinical conference or you go to publish a clinical paper, they ask you about your conflict of interest. In my case, I say none, because the conflicts of interest they're talking about is, do I have a direct commercial interest? And I don't. However, we all as scientists and researchers have conflicts of interest, because there's a outsized importance on our careers and our future funding on being the first to publish a novel result and being able to publish a lot of novel results quickly. What this means is that it sets up intrinsic biases for things such as mining data for expected results based on the dogma in a field. That it's actually well known that if you get the result that you expect, people are way less critical about that result than if you get a result you don't expect. And this is true at individual labs. It's also true even when you go to publish work. Also, we all like to be right, so hold on to our hypotheses too long. So you think you know what's going on. You get a piece of data that tends to discount our hypothesis. The first reaction is that that piece of data is not valid in some reason. The mistake isn't our hypothesis, the mistake is the data. And there are many, many cases of graduate students coming to advisors saying that I did not get data that replicated the hypothesis, and the advisor saying, it has to be wrong, do it again. However, if that graduate student had come and had actually gotten the expected result, that would not have been the reaction of doing more and more replicates. So this can cause a bias where you basically are reporting data that in a selective way. There's also a reluctance to report and challenge the results reported by others in the field. That there's this perception that if you get data that's against somebody else in the field, that the person who collected the original data will basically be offended, or there could be bad consequences to you. And I'm not saying that this doesn't happen, but fundamentally that I can think of many examples of somebody getting data that is directly counterdicts something that has been published. And either they don't publish it because it's too hard to publish it, or they don't publish it because they don't want to rock the boat of the field. And then finally, there is selective and non-rigorous reporting of data. That is the outlier effect, is that there are actually direct statistical tests on how to remove outliers from a data set. However, if you see, it's very much human nature to say, oh, this would be statistically significant if there wasn't that one piece of data that was outlying, and we're just going to discount that. And that really can skew results if that isn't done rigorously and carefully to discount outliers in data. Now, finally, it's that as humans, we're wired to find patterns in data, even when they don't, in fact, exist objectively. These are, I think, a really big one that is a good example of this is when we have déjà vu. We basically are like, oh, I dreamt something happened during my day. I dreamt that last night. And that's very possibly that's true, but there's not a rigorous collection of data of everything you dreamed, what percentage of it actually came true. And this is true when people look at data. I can't tell you how many times I've heard people say, well, it's not statistically significant, but the trend is in a certain direction. That is the whole idea of finding patterns in data, but they don't exist objectively. And getting people to basically say the trend doesn't matter because it doesn't exist objectively is a very, very hard thing to get people to do. And this is an example that is in a lot of discussions, and this is this whole idea of what people call PHacking. And that's manipulating, and it could even be unconsciously. It's not really meant, it's not if people are trying to be dishonest, but they don't realize how problematic it is. So basically, you're trying to get a 95% confidence interval. So you miss your confidence interval. Well, maybe if I used a different statistical test, that would give me the right 95% confidence interval. Another thing that can happen is that you think you know the number of biological replicates you need to be able to get statistical, to be able to look for a statistical relationship. And that's what your power analysis says, or that's what you think is true based on prior information. But your p-value is 0.065. And you're like, well, if I look at this data, there's a lot of variance. Maybe I'll just do 2 or 3 replicates, and that'll get me to statistical significance, and then I can publish it. Now, this can actually be problematic and directly lead to irreproducible data. But it can also give us new insights. So it's a situation where we have to be very careful, because the good part of this is that it can reveal new unexpected insights from data collected for other purposes. However, it can fool researchers by finding relationships that are occurring in the original data by random chance. And this is fundamentally the problem with the multiple comparisons problem. I've never really understood this concept when people explained it to me until I started doing a lot of high throughput genomics data. When you're doing high throughput genomics work, especially when you're looking at gene expression, you're basically simultaneously looking at the expression of 25 or 30,000 genes. And if you're using a p-value of 0.05, that means there's a 95% chance that the two things are different is real. But the other side of it is that it means there's a 5% chance that the relationship you're looking at is not real. And that chance is fine if you're only doing a couple measurements. But if you're doing 30,000 simultaneous measurements at a p-value of 0.05, a very large number of things that are coming back as statistically significant are in fact due to random chance because you looked at 30,000 relationships. And under these circumstances is that this is also a problem that when you collect the data set for one purpose, and then you start looking at it and start throwing statistical tests at it, in the end, you've done so many comparisons with the same data, you can start getting statistical significance that can never be replicated. Okay. So with all of that, NIH really needed to respond. There was a lot of bad press. It was really, showing up in Congress. Congress basically reacted saying, well, if all NIH research is useless, why are we putting so much money into it? So NIH has responded in 2 ways. The first is that they provided funding for training modules to improve data reproducibility. And this is a website that has a number of training modules to enhance data reproducibility. Some of this is really related to important statistical treatment of data and how to avoid the multiple comparison problem, how to make sure that you're not not reporting 95% confidence intervals that have been manipulated. Now, but the second, which is really what the topic of this presentation is that they also changed the NIH grant application and review criteria to ensure that the best practices are being funded for followed grants. Now, what this really comes down to is that we as investigators, they've just made writing our grants even more harder than before, because we have to address a whole lot of other issues explicitly that we didn't have to before. Now, the first one is we need to now, and the reviewers of the grant are now asked to specifically evaluate the scientific premise of your work. They're also asking us as investigators to specifically address in our research plans what the scientific premise is. And this means explicitly talking about the strengths and weaknesses of the prior research used as the basics of the project, this is both your own preliminary data, and under those circumstances, that can be relatively easy. You're saying, I did a pilot experiment. I now need the money to do a whole lot more replicates. In that case, you're actually saying that your preliminary data is hopeful, but the weakness is we need to do more replicates. That's what you're asking for money for. The tricky bit is that they're also asking you to describe the strengths and weaknesses of the preliminary data in the literature that are not your own. And that, so if you are saying that the literature has said that this is what's going on, and so I'm going to be looking at this aspect of it, they're asking you to specifically say how good that study was. How were the replicates? Has it been replicated by others? Is it well designed? Is there a potential that that preliminary data was not reproducible? Now, under these circumstances, they're asking you to address how rigorous is that prior work? Is there reasons to doubt that reliability of that preliminary data? And does your proposed research address the weaknesses and gaps in the scientific premise? Now, when you're using this to address gaps and weaknesses in your preliminary data, that's really no different than we've ever written a grant application. That's saying that, all right, I have this preliminary data that suggests something is true. I'm now asking for the money to really rigorously validate it. However, the tricky bit is trying to address the weaknesses of others' data. And that this will require being very, very politically astute, because it's possible, if not likely, that people involved in doing the other data could even be involved in the review of your grant. So you have to be very careful to, just for human nature, be addressing this in such a way that is saying that that this is probably true for these reasons. If you think that there's reasons that it could be somewhat unreliable, be careful on how you pose that, because yes, you have to point it out, but on the other side of it, you want to make sure you don't offend people. And this is something that, in my opinion, NIH has not adequately addressed when they put this criteria together. Now, some things that you do need to specifically address, and this is things such as that you're basing it on a small study with few measurements. In my field, you could be basing it on a study done on a transformed cell line that may not reflect in vivo biology. If the effect size is small, maybe there's a 10% difference between normal and manipulated. That may not pan out when you go and do it again. A study with large numbers of tested relationships. This is an issue with these very, very large multi-center studies, such as like the nurses study, or where you're looking at things going on with 10,000 people, and people are throwing you're doing all sorts of comparisons that, again, you're going to have that multiple comparisons problem. So are the effects that are being seen, are they actually real? Was there iffy experimental design? Did the study have possible financial conflicts of interest, such as a study performed by a drug company? Or a study that's very exciting but has not yet been independently replicated? Studies that had iffy experimental design, if you really feel that way, be very careful about using them to justify your scientific premise in the first place, simply because if you don't want to be pointing out that the studies underlying your proposed work are in fact poorly performed. So this is actually a reason to really make sure that the very best design studies are used to underlie your scientific premise. Now, the second review criteria is scientific rigor. And this also must be something that you have to specifically address in your research plan. And how NIH defines that is the strict application of the scientific method to obtain robust and unbiased experimental design, methodology, analysis, and the reporting of results. The problem here is that means that you as the investigator are expected to more explicitly outline your experimental controls, your plans to reduce bias such as blinding, your inclusion and exclusion criteria, your power analyses, and your statistical methods. Now, many of you may already have to address these topics explicitly in your grant applications, and this is really true in much clinical research. In my field, where I do a lot of preclinical stuff, that we never would explicitly outline our experimental controls and grant applications up to this point. We wouldn't talk about blinding, and we wouldn't talk about power analysis statistical methods very much. It would be a very, very short inclusion. Now, though, for all applications, reviewers are being asked to specifically look for these things. And the problem here is that we have to specifically talk about all this additional stuff when we're already working with an extremely short grant application. So 10 years ago, the NIH grant application went from 25 pages to 12 pages. That meant that much of the information that we used to include, such as experimental controls and so forth, there wasn't room to address it. Now we have to include this stuff back in the application with the same page limits. So this is why this can be very, very difficult to actually deal with. Now, the other issue that we now have to explicitly address is the consideration of relevant biological variables. And this is something where we need to talk about how different experimental approaches could change our result laboratory to laboratory. If we're talking about cell lines, this could be passage, or how long that cell's been in culture. Where are you getting it from? Are you getting it from another investigator or a cell line repository? The difference between a tissue cell line from a hamster versus a human versus a mouse versus a rat, exactly which cell line you're using. For instance, if you're studying glioblastoma, different glioblastoma cell lines will have extremely different behavior. So why did you pick that cell line, and how is that of value? If you're working with experimental animals, the genetic background of those animals, because there can be huge strain differences in behavior, immune system function, hearing, sight, and et cetera. So in the past, we wouldn't actually have to specifically address why a certain genetic background was being used. Now we need to talk about how genetic background could affect our results and how we're going to control for that. And genetic background can be so severe, even the vendor you buy your animals from can matter. So how are you going to control for that? Your housing conditions of your animals, how age is a biological variable in animals or humans, weight, body size, or diet. For human studies, how does socioeconomic status play in, or underlying health conditions as well. And while sometimes we would address these priorly, now the reviewers are asked to specifically say, how well did you address these issues? So it's putting reviewer attention on things that often people would get a big pass for in grantsmanship. Now you have to make sure you talk about consideration of relevant biological variables. Now, a really big one, which is extremely important, is also making sure that you specifically consider sex as a biological variable. And the reason for this is that historically, male animals were used for many, many studies because they wanted to reduce variability that could be caused by the estrous cycle. Also, almost all clinical trial drug studies were done on adult men. And the reason for this was, again, women were considered more biologically variable. And also, they didn't want to have the drug done on a woman who could be pregnant. And if the drug has a teratogenic effect, they didn't want to have to bring that up. So what this means is that drugs are approved for use by their interaction with young male physiology, because also older people weren't used for these studies. There are plenty of cases where drugs that are metabolized well by young people are not metabolized well with old people. Also, most drugs on the market have not been directly, specifically tested with female physiology. Many cases, that's fine. However, if there is a bad drug reaction with female physiology, then there's a real problem. And it also means that underlying causes of known sex-specific differences in disease incidence and pathogenesis are largely unexplored. There are sex differences in onset of cardiovascular disease, of cataracts, of multiple sclerosis, all sorts of things. But if you're only studying men, that you can't get a handle on what those sex-specific differences are. So this consideration of sex as a biological variable also must be specifically addressed in the research plan. Now, NIH claims that this does not mean a study needs to be powered to detect sex differences in the result. And they're saying this because it means that this is not sex differences research. However, the expectation is the default is that all studies should include both sexes in a study. And awareness is maintained that that introduces a biological variable in the experiment. However, so you're looking for, so the idea is to be aware. If you see trends, then maybe to go back and redesign a study to be powered sufficiently to detect sex differences. And I want to emphasize that this is a very tricky place to be. And there's a number of, in the papers that are up on the site that are supporting this presentation, there's several talking about this issue of how to deal with sex differences as a biological variable. Interestingly, and something that is really under considered, is even sex differences in cell lines, because cell lines are either coming from male or female animals or people. And how do you actually control or think about sex as a biological variable when all your cell lines might be male derived? And this is actually specifically addressed in one of the papers that's attached to the presentation. Now, in the end, that if you really want to only use one sex, you have to specifically address it and you have to specifically justify it. There's really only two acceptable reasons to only study one sex. One is that if it's a phenomena that only affects one sex, such as prostate cancer, that people have done the real sex differences research to show in a rigorous, convincing fashion that the phenomena has no sex-specific differences. The thing that NIH is emphasizing is that cost or convenience are not acceptable reasons to study one sex. So while they're saying that the study does not need to be powered to detect sex differences, like in my field, that it can be very expensive sometimes to study male mice because male mice have to be housed at a lower animal housing density than females because they'll fight and they'll be aggressive. So I can't propose to only use females because using males is inconvenient because of housing considerations. So from that perspective is that they're basically saying is cost or convenience are not acceptable reasons to only study one sex. So as I mentioned earlier, the bad news with all of this is that the same information needs to be included in the same page limits as before. And what this really comes down to is that it's even more crucial to write your first drafts early. And when I talk to people and students and many people will start writing a grant writing to a page limit, which means is that you are saying, I can't put that in because it reaches my page limit. I can't talk about this because it reaches my page limit. And in the end, you might be writing a grant to a page limit, but it doesn't actually address all of the information that the grant requires. So what I would recommend to everybody is that you sit down and you write your initial draft of your grant with no regard to page limits. The most important thing is to make sure all of the information needed is included in the application. Now, this might mean that the first draft of your R01 is 20 pages instead of the 13 that you're going to be submitting. But I don't know, but many, many people, and myself included, tend to write first drafts extremely wordy and repetitive. Once you have all of the necessary information on a page, then you start wordsmithing. You start removing redundant languages. You start saying, instead of the tendency to say, it has been shown that, almost always you can collapse that to it is. And that's really the strategy you're going to have to use to get your grants down to page limits, which means is that you're going to need sufficient time to get everything down on paper so that you can brutally edit it down to the NIH page limits. And if necessary, then that's also the opportunity to get other people to read the application. And at minimum, you definitely want to get feedback on what's in the application. But one place where you can get that feedback is to ask other people for help editing for length. What excessive verbiage can you get rid of? What things have you said 4 times that can really be brought down to 1? So from the standpoint of scientific premise and scientific rigor, those are considered scorable parts of the research plan. They're also scorable things that NIH reviewers are now being asked to directly put into their final impact score on your grant. Now, there's another part of the application you do need to write. It's not a scorable criteria, but it must be included if the grant is to be funded. And that is authentication of key biological or chemical resources. It's an additional separate 1-page section that goes in the application. This is not a place where you have room or they want you to elaborate on your experimental plan. Which this is specifically designed for, is how can you prove you know what you're studying? From the standpoint of if you're working with animals or mutant animals, you have to provide information on how the mutation is ensured. How is the genetic background determined and maintained in a long-term breeding colony? And how are you controlling for genetic drift? If you're working with a cell line, how will you ensure that the cell line is what you think it is? And how will phenotypic drift be addressed? Because even if your cell line is validated, each passage is going to cause new mutations in that cell line. And how are you going to deal with that drift of phenotype that occurs in cell lines? Now, there's some good news for this, is that Delaware Embry has core facility waivers that are specifically designed for validation of cell lines and genetic background of animals. And so you can go to the Delaware Embry core waiver page and actually request money for this. And it's this new money that came from NIH. So if you think that you're in a situation where you want to do this, definitely apply for that money. I think it's available through March, if I remember correctly. Another place is antibodies. If you're using antibodies, how will you confirm the specificity? And the gold standard is using that antibody in a situation where the animal does not have the gene of interest. That's often not possible. So they're going to ask you, how do you make sure that that antibody staining is real? They're going to ask you explicitly, are you doing peptide blocking, Western blotting, confirming molecular weights, and so forth and so on. Also, if you're using specialty chemicals, let's say you're buying purified proteins, let's say from Sigma Aldrich, or you're using inhibitors that are chemically synthesized, how do you know what you're studying, what you think you are? This will involve things like validating purity, perhaps doing a Western blot yourself to make sure what's in the tube you're buying from the company is really what you think it is. And this is something that needs to be all outlined specifically. So from the standpoint of it's not considered in scoring, so why put a lot of effort into this? This is the same, you put a lot of effort into this section, the same reason that you need to put a lot of effort into your human subjects and your animal subject sessions of your grant. Why they are often not technically scorable sections. It's very, very important to address them, because if you get a fundable score, but those sections are not acceptable, then the grant you'll be asked to then provide acceptable information to the NIH. The problem is that if it's acceptable during grant review, the section is almost never looked at again. If it was unacceptable in grant review and you're going back and backfilling, it is much harder to get someone to sign off that the section is now acceptable. And I know of many cases of grants whose funding was held up for weeks or months. And in one case, I even know somebody who ended up not getting their grant because their original animal section was considered unacceptable, and that the people who actually checked it at NIH never would approve the revised section. So this is a situation where if it's fine out of the gate and the reviewers say that what you did is fine, then you're good to go. So you do not want this to go to the NIH level review if you can't help it. So it's a very important section to be addressing. So with all of that, is that the take-home message here is that there is a new set of guidelines for all NIH grant applications that are going in after February 1. There's an entire new NIH grant guide, and that you should, if you're working on grants now, you should be downloading that. One of the things that includes is that all new grant submissions must specifically address the issues of scientific premise and rigor. Now, I think we all can realize that in the past, good grant writing addressed these issues to the point where the reviewers had no concerns. But often this wasn't explicit, and reviewers often would give investigators the benefit of the doubt. Of course, they're doing this correctly. However, now it's not just you're given the benefit of the doubt. You have to address it explicitly in your application. I would highly recommend you use the NIH nomenclature for scientific premise and rigor. So actually talk about the scientific premise of the application this is the strengths and weaknesses of the scientific premise, and use those terms. It makes it easier for the reviewer, and you know then that you have actually addressed it. That you have to specifically address the biological variables in your experiment and how these biological variables are being controlled for. And you have to develop a new section of your application, which is your validation of key resources. Now, many people who are listening to this or on this call may not have key biological resources. But if your study uses no key resources, you should still include the section, but just state that the study does not use any key resources. Because what you don't want is to have, again, the reviewer say, oh, the section's not included, when the issue really is there weren't any key resources for your study. So with that, I think I am done talking. And if anybody has any questions, I will be happy to address them. Or if there's any topics that you want to see specifically talked about in future presentations, please let me know.
Hi, thank you for the talk. Can you hear me?
Yes, I can.
Okay, I have a question about the scientific premise and rigor. So I thought that those two are two separate things. I thought the scientific premise was the knowledge gap and rigor was about reproducibility. Is that not the same?
It's not. It's the premise is not actually the knowledge gap. The premise includes the knowledge gap, but it's also that how good is the studies underpinning what you're doing. So, for instance, like you can use the premise to point out where the knowledge gap is, but it's also how good are the preliminary data. And that preliminary data could be your personal preliminary data, or that preliminary data could be other studies from the literature. So I think what you're getting at is like a study that we published recently is that the problems with a prior study that somebody else had published is what we had used as our justification for doing our study. But but it's also that very often you'll say, we think this is happening because these 5 other papers have shown this in, say, in another system, or that we believe that this is the biological mechanism because, like in my field, I study wound healing after cataract surgery. And one of the big scientific premises is that this is very, very regulated by a growth factor called transforming growth factor beta. Now, the scientific premise there is actually excellent. There is probably 100 papers showing that this set of proteins regulates this process. So if I am proposing to study how something I'm looking at is affecting this transforming growth factor beta, I have an excellent scientific premise. It's extremely well established. But let's say I am looking at another signaling profile like HGF signaling. There's one paper in the field that suggests this HGF signaling could also be important. The scientific premise for that is nowhere near as strong. It hasn't been rigorously replicated by other people. So there's a difference between the thing that everybody agrees on because it's been replicated in 20 labs 100 times versus the one study that maybe was using a transformed cell line. And while it's indicative, it's not definitive. And part of this premise is to point out is that if you're proposing to study HGF signaling, pointing out that we may not know as much, that it's not as absolutely certain that HGF is important in the process. So
Okay.
it's a little, you see the distinction I'm making?
Yeah. So scientific premise and rigor is just one thing that we have to address. It's not two separate.
Well, the thing is that rigor, there's the rigor of the scientific premise, but then there's also the rigor of the experiments you're proposing to do. So when you're looking at the scientific premise, part of what they're asking you to address is how rigorous were the studies that underpin your work were. And is there an issue with the rigor? And is that something you need to be addressing in your new experiments? But there's also the issue of how rigorous is the design of the new experiment you're proposing. So things such as, have you done your power analysis? How many biological replicates are you using? Have you adequately validated? Are you making sure you're considering your biological variables? And this sort of thing. So rigor comes into it in both the stuff that happened in the past as well as the stuff that's going to happen in the future.
Okay. So specific sections that you would recommend putting in is the rigor of the scientific premise and validation of key resources and accept as a biological variable.
Yeah,
and
so
the future study is not going to be a specific section, is it?
Well, the thing is that a lot of this might not even be specific sections. So when you talk to there's the part where you basically set up the field. You might be talking about scientific premise there, where you're setting up what the scientific question is. There you might be specifically addressing scientific premise about how validated is and how well accepted is what we think we know is. And then you also might be bringing it up again because you might be talking about it as you're justifying your individual experiments, because it's a mistake to put all of the background in the introduction part, and it's the background and significance part, and it's a mistake to just put only experimental design information in the experimental section. And that's just because the reviewer won't be able to follow it as easily. So if you are proposing an experiment and you're saying that I'm proposing it because this piece of information is known, you might be talking about the scientific premise of that piece of information locally in the research plan part. So you probably, you're not going to be making necessarily a separate section addressing these things. It's more when the topic comes up and is relevant, you address it right there. Does that kind of make sense?
Thank you.
Is there anybody have any other questions for right now? Okay. So again, I encourage you to let Erin know if there's any other topics you want addressed, and that we can go from there. So everybody have a good holiday, and we start up with the intro to grant writing ones in the new year.